is already time to start the session. So we'll begin the session in the afternoon. Assalamu alaikum and very good afternoon. In this session, we are going to have two speakers. And the very first one is Dr. Imran. He's an associate professor from Bahauddin Zakaria University, Multan. Dr. Imran has received his AMPHIL from ICCBS and later on he proceeded to Goethe University in Frankfurt, Germany on the DAD fellowship. So he is basically a DAD fellow as well. And after completing his PhD and on his return to the university where he is serving as an associate professor currently, he has this he established a first EEG lab in Pakistan in his university, and in addition to that, he also received a donation to establish an automated lab for the behavioral study, and he's received this grant from King Saud University in Riyadh, and these are his initial, very beginning achievements at his university. Basically, his interest, Dr. Imran's research interest is in the field of neuropharmacology, molecular and cellular pharmacology, and with special interest in the epilepsy and its related models, animal models as well. The title of his presentation is Pharmacological and Behavioral Evaluation of Fear Reducing and Memory Enhancing Effects of Phyla in rats after a UHPLC MS analysis and incident-based studies.
Thanks a lot, madam, for your nice introduction. Thanks to my mentor for basically I have learned so many things from madam at that time. So Dr. Sheikh Ashraf Sir, madam, and Dr. Asna Dor. So basically this is the foundation and seed I learned from here, and then later on keep executing the things. So I'm apologize for the wrong title. Basically, I asked my master student to send me the updated slide, and she actually posted the title of the publication, so which has been just published in the article. So anyhow, objective is the same. So basically, I mean, the work is basically based on the use of the native plant and on exploration on its behavior and then oxidative stress parameters. So, and this work is affiliated with the master student. And in this case, the work has been basically done on the assessment of the memory function with the use of some the latest equipment. I was in Germany in 2014 when a Nobel Prize has been awarded to Edward Moser and maybe Moser on the hippocampal space allocation. So I had an opportunity to meet them, and at that time I thought when I would be back, I would apply epilepsy and behavior and memory learning part with my own reporters. Basically, Alzheimer's disease, you all know, it's a burdenized situation. I mean, as the age is progressing, particularly in advanced countries, in Japan and in Germany, in Switzerland, because the life expectancy is over there about 80, 90 years of age, and Alzheimer's disease is a problem. People are in the area of the dementia kind of thing. So basically, natural products, I mean, it's a continuous momentum story that people are talking about it, that either we should use natural products or not. It's a continuous debate. Anyhow, in this my case, I asked my master student to use Phyla nodiflora. It's a native, it's a fruit and being used for purpose of soothing and calming effect for a local community of the southern Punjab. So I asked to investigate on the scientific basis. Basically, my German university or my collaborator had a belief that if healthy, your brain would be healthy. And 50% of your troubles of your brain would be gone with healthy food, with the healthy molecules. So basically, with the healthy food, healthy stuff, polyphenolic compounds, and this kind of thing, VNF is going to high, inflammation is going to reduce, that kind of thing. And based on this, recently I published a paper on the falsa, basically Grevia asiatica. It's a native plant of my Multan region, and I published article in Frontier in Nutrition based on, and I never use extract. It's a fresh juice. I administered the mice and rats, and later on and demonstrated the improvement of memory function. So my target goals were basically the implication of this plant on the assessment of the fear and then assessment of the memory function. And then subsequently, I sent the sample to King Saud University to my collaborators for the purpose of chemical characterization and then in silico studies. And then also for the drug variable variability process. So study protocol very simple, extraction and then subsequently in vitro screening with a very basic assays for the phenolic content or the flavonoid content, DPPF and ABTS. And subsequently, then this is the actual protocol which I adopted for which is a chronic study. So actually it has been administered from day 1 to 30 in continuous administration of a sphalanodic flora and then after subsequently 30, then the behavior assessment has been done on the males for Zolivec, open field, light and dark, elevated flux males, Y males, NOR, and Morris water males. Basically, least aversive to maximum aversive approach. And these are the groups basically. Some of them are controlled, some of them are positive control, and then some treatment groups. And these are the behavior studies, behavioral plans I have used. In anxiety cases, the open field test, light and dark test, elevated plus males, and test for learning Y males, non-object cognition test, and Morris water male test. And biochemical assays, it has been performed with the help of biochemistry department in my university. So results, the results are protein content is milligram 27.27 milligram of quercetin equivalent per gram, and then gallic acid and the IC50 value for the DPPF assay and ABTS and stanford assay to display here. And then I got this data from King Saud University HPLC analysis, and then subsequent demonstration of different compounds, which has been later studied. So this is the open field test. I'm using basically automated system. Open field is completely integrated with the software. Everything is assessed on in real-time basis. And then animal exploration in the central zone and animal exploration of the peripheral zone. Because animal has natural instinct that it try to avoid. If the animal is afraid, then it try to be a thigmotaxis behavior. It's a kind of the behavior animal remains surrounded the ball. So this is the case, and then these are the simple two parameters I observed: time spent in the central zone and time in the enters in the central zone. So if animal is in anxiolytic situation, then definitely it tries to explore the region. So these are the outcomes. Of course, diazepam is the standard drug, and we use very low amount of diazepam, only 2 milligram per kilogram of the diazepam, because it has less sedating potential. Otherwise, increasing the dose has huge impact, and we will like with the sedation. And then you can see the effect that as the dose has been increased, dose dependent pattern of anxiolytic has been observed. And light and dark box, this is again software integrated. Everything is being animal is performing in the open, in the light and dark region, but the transitions are being calculated by the software, which is basically Athroion Nodus. And you see that chronic usage has demonstrated the pattern very similar to the diazepam. This is the light and dark box. And then elevated plus male, this is the equipment we have used. And again, the animal has instinct. If it has calm effect, then it prefers to be in the open area, in the open arm. In this case, the 300 and 200 milligram per kilogram demonstrated that animal actually is more explorative behavior in the open arm. And that's for learning and memory. In this case, I use Y males and percent of spontaneous alteration. This is the parameter I have noted, that percentage of alteration. And then scopolamine. In this case, we have challenged the mice with the scopolamine. 1 milligram per kilogram, because this is the amnesic drug, and animal it goes into the amnesic situation. And with paracetam, initially I have used paracetam with the scopolamine with the concept, with the intention of nootropic drug. Where in the later studies I have replaced this molecule. Now I'm using this Stanford University. So, and in this case, paracetam has very good effect, and certainly it has no mechanism explored as yet. And the combination, it has antagonized scopolamine induced amnesia. And now objective cognition test, this is another test I have integrated in my study. And in this case, the discrimination index has been calculated, it has been demonstrated, and those animals, basically the treated animals, they have demonstrated some better discrimination index with the progression of treatment. So this is the case with Morris watermaid I have used in my studies. So Morris watermaid basically it is a very well-known method. It has very good protocols by nature method in 1984 paper, and this is very strong actual test for the assessment of the place preference and then reference memory. And in my case, I have modified the protocol a little bit because what I have figured out, because many of my colleagues are in discussion with me, in this case, I actually use distal cues and then, sorry, proximal cues and then distal cues. So I gave the two cues to the animal, means proximal cues, and in my observation, so what I observed that animal actually explores more than proximal cues. And based on the proximal cues, then it makes the coordinates, it makes the orientation. So my platform zone was actually southwest zone, and then we explored basically software integrated system again. And what I have figured out that, so basically day one, day two, I omitted, it was just a training day, because animals just, I mean, we trained them for the stabilization of the equipment. And then you see the scopolamine treated animals, they remained in the stable latency is quite high. And they were not able to locate the platform even under the impact of scopolamine. So a treated animal and parasitic animal, I mean, they were quite efficient and they located the platform very efficiently. And track plot, it has been generated by the software, and distance traveled, I mean, the coordination. So this protocol has been done for 5 days. Day 1 and day 2 is the training day, and then day 3, day 4, day 5 is the experimental assessment day, and day 6 is basically probe day. So next day, we have completely eliminated out the platform. The platform has gone, and now there's no platform because we just want to see the memory because it's a natural instinct of the animal. It needs a platform for its rescue. So in this case, the platform is there's no platform, but animals try to spend maximum time in the region where the platform is actually. So it's basically a reference memory. So in this case, again, the paracetamol, no doubt, is the best, but some treated animals, somewhat they demonstrated that animal is trying to locate the platform. And biochemical acid basically means MDA acid or dialdehyde acid, lipid peroxidation, peroxide dismutase, and glutathione peroxidase, and usually used for the assessment of the oxidative stress in the brain homogeneous. And this job has been done with the collaborative part from the Department of Biochemistry. And what we have seen that the treatment has restored many of the oxidative, pro-oxidative, or oxidative, sorry, or antioxidant enzyme restoration. And then this is the chemical. These are the chemicals, molecules I got from the UPH, ultra high performance liquid chromatography. And then these compounds basically based on blood-brain barrier permeability, we have selected 4 compounds, and then these were sent for the inductive studies basically to assess, are they doing any job related to the acetylcholinesterase activity or not. So based on the blood-brain barrier permeability, and then so these are the some outcomes. And so in these compounds, basically they are targeting the acetylcholinesterase and fitting in the body target, and then inhibiting the acetylcholinesterase and probably increasing the acetylcholine level in the case. And this is another case. There are another 2 molecules. And overall conclusion is that plant is, of course, it is all the time it is claimed that they are rich in phenolic compound flavonoids. So basically, this by analysis, they detected a 22 compounds. Among them, 6 of them they belong to the flavonoids, and 5 of them they belong to the alkaloids. And basically, 3 of them they have been selected for the doctoral studies and then sent for the acetylcholinesterase acid, and eventually it has been figured out. So basically, it may be presumed that combating the oxidative stress level would be the responsible, because there are numerous studies done by the many renowned scientists that scopolamine it actually increases the oxidative stress level in the brain due to the different environment, maybe receptor occupation and then problematic situation. So maybe antagonizing, combating the situation with oxidative stress would be a possibility. So that's the case. And acknowledgement to my PhD students, my master student, and my foreign collaborator, Dr. Paul Kaplan, to whom I'm working since 5 years. So basically, he's the expert of microdialysis, and I also did microdialysis together with him in Frankfurt. And this work has been done by master student Talha Farooq. So that's a part. So So right now, my lab is basically involved with the EG studies. So in coming days, in coming months, we'll be able to publish inshallah those works also. That's all from my side.
Thank you, Dr. Imran. It was indeed a very concise and very informative, interesting lecture that you have delivered. Since you have finished well before your time, allotted time, so we still have 5 minutes for the question.
Okay, ma'am, thank you very much for the nice presentation. I don't have that much experience with the behavioral studies. I just want to know that when you do these behavioral studies, do you also see the expression of some genes or not?
So at that time, this work has been done in 2017. Recently, it has been published in Journal of Physiology and Pharmacology. Last year, 2021. So at that time, I'm following the German formula step by step. So right now, I have incorporated a real-time PCR facility at my site. So it is in the phase of optimization. I told Madam 2 years before, I got the EEG setup. Honestly, 2 years gone. I'm still in the phase of optimization. So I have this facility right now, but I haven't done any gene expression or this kind of stuff.
I have another question. For example, like when you do such behavioral studies, you just conduct all your experiments on the one generation, I think, like we call the founder mice or founder rats. So what about, like, for example, you have already established this protocol in just one generation. So we will not have another generation. For example, if you want to study Parkinson's disease or Alzheimer's disease. So how can we justify that just lots of energy, lots of financial in this one generation?
I use spot rats UV. Basically, there's another species, Vista rat, long Evans rat. It's variations everywhere in terms of the behavior. I have to agree. But recently, my colleague in the next hall is going to present another paper, which is basically more advanced. He worked with the epilepsy model, and in that case, we induced the disease. And then we assess the behavior studies. Basically, in my opinion, in mice, if inbred or outbred, there's a huge variation. So, maybe the behavioral people could better explain also that even a single stimulus, because we in Multan, we sometimes face a problem of the light outage. And then next day, we see completely disorientation in the behavior of the animals. There's a strong impact of the changing of the strains. But what I know in the case of epilepsy, I could let you know a little bit that I have evaluated the maximum electroshock seizure in the case of in Germany, in Frankfurt, in CD1 mice, and then BARC mice, and then LEJNSIC mice, and then Swiss mice. And in all cases, I found the different EC50 values. Yeah, scopolamine was administered with the memory test. And it was administered at the day of experiment, acutely. Because there are a lot of studies. If we use scopolamine for the long duration, then it has evident effect on the brain functionality, brain network rewiring. It's a very phenomenal it has been used. And neurogenesis people could also know.
Are there any other questions?
Sir, I want to ask one question. Memory resides outside the brain. Is there any method to assess the memory that resides outside the brain?
With animals or?
Like animals, in the animal model of study.
In Germany, just a brief story to let you know. In Germany, one rat had survived impact of my status epilepticus. And I kept him alive for 1.5 years. So I kept his name Polly. So as soon as when I entered in mother, I just called, "Oh, Polly, where are you?" It became anxious because every day I gave him a chocolate and toffee and biscuit. And subsequently, when I entered his lab, it became anxious that Imran is in the lab. So I just offered one day, come and sit on my shoulder. And there's a little Stewart movie, it behaved exactly the same. It remained on my shoulder for more than 2 hours, very calm. So, rats, in comparison, rats have very strong memory. But I have to agree, mice, they have, they don't have consolidated memory. Mice forget after some time, but rats, they don't forget. That's why Edward Moser and maybe Moser, they use rats log evens rat in their memory studies. I mean, rat, they are friendly.
Thank you.
I must say that if you have any more questions, Dr. Imran is here for a few more days, so you can interact with him and ask him and discuss whatever you want to on this. Dr. Imran, you have this
In the abstract book, there's an email, you could write any email.
sculpture presenting this on the behalf of the organizing committee as per the appreciation. Okay.
I appreciate it because I learned, this is my institute where I learned a lot. And then later on,
So
honestly,
this is alma mater, ICCBS, this is the alma mater.
so when I was in a DOD interview in Germany, Frankfurt, Bonn, they asked me, from where did you master? I think I did my master from ICCBS. I think it was one lady, she was from Germany. Oh, then there's no need to ask, you are adequately trained. That's it. Thanks a lot.
Okay, with that, we are going to proceed to our next speaker, Dr. Muniza Sheikh. She is a research officer in Dr. Kandwani Center for Molecular Medicine and Drug Research. Dr. Muniza completed her PhD in 2019 from PCMD and ICCBS, and under the supervision of Professor Dr. Iqbal Chaudhary. Currently, Muniza is working as a research officer, like I said earlier, at PCMD, and she already authored 13 international publications. She has investigated the inhibitors of protein glycation through in vitro biochemical assays. She got her training in the field of structural biology using NMR at University of Hamburg, Germany, under the supervision of Professor Meyer, where her focus was studying the protein-ligand interactions of therapeutically important enzymes using in silico designing of inhibitors, molecular docking, and dissimulation studies, and STD and NMR techniques. Dr. Muniza has been trained on the NMR spectroscopy, evaluation of kinetic parameters of therapeutically important enzyme, to progress curve analysis, estimation of binding epitope and affinity of ligands by saturation transferred NMR spectroscopy. She has been awarded distinction during her academic years in BSc Honors as well as Masters. She was also awarded Syed Abid Ali Zali Gold Medal for securing first class position in Masters in the Department of Biochemistry, University of Karachi. Today, the title of her presentation is In Silico Identification Potential Inhibitors of key SARS-CoV-2 3CL hydrolase and flow.
Assalamu alaikum, very good evening, and thank you so much, ma'am. It's always an honor to be introduced by your teacher. And so the title of my presentation today is In Silico Identification Potential Inhibitors of key enzymes, that is SARS-CoV-2 3CL hydrolase and flow. So, as you have heard that I have mostly trained for in vitro assays, but I have conducted this in silico studies right at the start of the pandemic in 2020, when the very first structure of this enzyme was reported in PDB on 5th of February 2020. So I was actually guided by my supervisors, Professor Dr. Iqbal Chaudhary and Professor Akil Wahab, that I should use our the database, the compound database that we have at our molecular bank, and use it for in silico screening against this important enzymes of SARS-CoV-2, which was deposited in PDB. So this enzyme, as you have, if you have taken today's lecture, invited lecture of Professor Atia, she has already explained that this enzyme is also known as a non-structural protein. It's because it has some catalytic functions, and having a name of NC5, and it have a significant role of cleaving the viral polyproteins and generating 12 non-structural proteins such as NC4 and 2 NC15 that includes the RNA-dependent RNA polymerase and the helicases that are crucial for their application and propagation of the viral gene. So inhibition of this enzyme or this 3-CL hydrolase would prevent the virus from replication and therefore constitutes one of the potential anti-coronavirus strategies. So that is why this target was also selected, not only because of its availability in the PDB, but also because of its significant role in the viral life cycle. So the salient feature of its structure is that it's a 3-domain cysteine protease, and it has the active enzyme is a homodimer that is containing 2 protomers. So the methodology that we acquired, first of all, the computational software that we used was Maestro Schrödinger, and the methodology followed was the preparation of the 2 PDB IDs from the protein data bank 6LU7 and 6Y2. Why we have selected, I'll explain it later. And we also did ligand preparation of the in-house chemical library of 15,000 natural synthetic compounds. Basically, these are more than 15,000. I just rounded off to 15,000. And then we did the in silico screening via molecular docking against these 2 targets of these prepared compounds. And then we do predictive binding energy estimation, and we also carried out there after the shortlisting of the compounds, we do ADME properties analysis, that is pharmacological or drug likeness prediction of these compounds, and we did some anti-simulation studies of 20 nanoseconds. So the selection of PDB IDs was based on, first of all, their availability. As I told you, that the crystal structure of 3CL Pro was released on 5 Feb 2020 on PDB with 6LU7. And we actually did all of our work and submitted it in the journal of our interest. And by that time, within few days, we got the revisions and we were asked to update our studies with 6/12, and it was just a month later, on 4th March, the next structure of this CCL Pro was actually deposited in PDB with a better resolution 1.95. So we also included that in our studies. Basically, we initiated our studies with this structure. Both have the difference of resolution as well as they have also the different compound co-crystallized in the active site. This one is crystallized with N3 peptide inhibitor, and this one is crystallized with alpha ketoamide. But the binding site is same. So first of all, we prepared the protein by protein preparation visit, in which we actually added the missing hydrogens that are lacking in the crystal structure and optimized the geometry of these compounds. And then we did out the 15,000 compounds that we have actually prepared through LIGPrep molecules. We converted them into from 2D to 3D structures, optimized their geometry, and you minimize them during the preparation. And then we carried out the molecular docking studies in you can say the extra precision mode of the glide. The glide is a module, the docking module of the Maestro, the docking software, and the results that we have obtained were the 9 compounds that were selected from the library of 15,000 natural and synthetic compounds against these 2 enzymes. The compounds, the selection was actually made on the basis of the docking score, and after that their predictive binding energies were also determined. And in addition to this, we also carried out some docking studies against the antiviral drugs, and they were also considered as the by WHO that these can be used as the remedy against the COVID-19 at that time. So we use these drugs as antiviral drugs as standard in our docking studies, remdesivir and imdevir. And compound from these 9 compounds, we selected actually 2 compounds, compound 2 and 3. And because of their highly predictive, negative predictive binding energy. These compounds gave interaction with these PDIDs. So from 6LU7 we selected compound 2, and from 6Y2F we selected compound 3. These 2 compounds also produce a good docking score as well as predictive binding energies as compared to this remdesivir and imdevir. As you can see, the docking score of with 6LU7 is -7.72 and -6.85, and imdevir has -7.59 and -7.10. From these 9 compounds, we, as I showed you that we selected compound 2 and compound 3 first on the basis of docking score and later on the basis of their predictive binding energy. And this compound 2 is of synthetic origin, and it was actually deposited by the group, Professor Dr. Khalid Khan, who is serving as a faculty here. And the other one is from natural source, and it is mainly found in Salvia mycorrhiza, Salvia family. And it was deposited by Professor Dr. Vikaruddin, who used to be the faculty here. So these are the docked poses or in molecular interaction of the compound 2 in the binding site of 6LU7. And the binding interaction was observed with glutamate 166, glutamine 189, and threonine 26. These were among the residues that were also found to be interacting with their respective co-crystallized structures in the PGID. And this is the compound 3 in the binding site of the 6Y2F, and here we can also see the interacting residues of threonine 26, 16, 44, serine 46, glutamate 166, phenylalanine 140, and glycine 143. So after that, the in silico ADME, that is absorption, distribution, metabolism, and excretion properties of these compounds were predicted in order to see, and we found that compound 2 actually fulfills the criteria to be a potential drug candidate, but the compound 3 has various outliers in case of solvent accessibility, solvent accessible surface area, and her donor hydrogen bonds and number of metabolites and percent human oral absorption. So that is why we actually suggested, or we actually proposed the further derivatization or further modification of this natural compound, so it can be acted as a potential or good candidate as a lead compound. And after that, the 20 nanosecond anti-simulation analysis of the compound was conducted, in which is presented here in the form of RMC, that is the immediate square deviation, in which the ligand profile or the ligand trajectory is actually overlaid over the protein backbone, and the fluctuation is noted. And fortunately, we had a stable complex prediction, and I mean, there are fluctuations, but they were of permissible range, not very much. And then after MD simulation, we also analyzed the difference in the dot pose and the changes that have been occurred, that could have been occurred during the course of MD simulation, but most of the interactions that we observed during the or after the docking were retained during the MD simulation analysis. And for instance, here you can see that this Renine 26, cysteine 41, glutamine 189, and methionine 65 tend to have the a stable interaction more than 30% of the allotted simulated time. And if you are seeing here, this is actually exceeding the 100% simulation time. It means it is making the same contacts of same subtypes at different percentages of the time. That is the cumulative effect of this interaction. Here, in the case of compounds, we have observed the same, you can say, the same number of interactions and same type of interactions as we have observed during the docking protocol. But we have also seen some new interactions also, but they were not of very much, you can say, significant, because they were not be persistent, or they were less than 30% of the simulation time. But other thing, this is also quite obvious because of its ADME properties, like it has the tendency of making or accepting 14 hydrogen bonds. So that is why we are observing this kind of behavior of this compound. And but since it has some poor ADME properties, so its further modification was required. So the study actually was published in 2020 and PLOS One with this title, and I would like to acknowledge the contribution of the industrial partners who have contributed in funding this research, and also contributing the compounds that were by the professors Dr. Vikarudin Ahmed and Professor Dr. Khalid Khan in the molecular institute. So with this, I would like to end my presentation here, and thank you.
Thank you, Dr. Badelza. I'm really amazed by both the presenters. Their time-keeping skills, because both of the presenters of this session has finished their lecture well before the time. So we have 5 minutes for Dr. Badelza. If you people have any questions.
Yes, Munira, I'm sorry that I missed part of your presentation. So this was for the epitope mapping, right? This is the interaction between the ligand and receptor. If you want to know exactly where receptor, the part of the receptor which is interacting with the ligand, which experiments you need to do?
Basically, sir, we are still planning to do it through biophysical techniques like NMR. NMR is one of them, because it has got the ability to do it through chemical shift mapping. Either we can do the ligand-based interaction or the protein-based interaction. So in protein-based interaction, we need to have a, you can say, the labeled protein. That is a difficult approach. So that is why we are trying to do a ligand-based approach, in which we will try to do the epitope mapping, as you have said. And the
NMR structure of this protein is not known?
But sir, the X-ray structure of this protein is known, and the NMR structure is actually being, we are trying to resolve it. Professor Athia's group is also, the protein is also expressed. It's in our hand, but the structural assignment is on the way. So hopefully we will be able to do these studies in the future.
Thank you. Shabash.
Thank you.
Any other question?
Okay.
Hi. Wonderful talk. Thank you so much.
Thank you.
I do have a question. In your RNA helps for the molecular dynamics simulations, can you bring this up again? I couldn't have noticed that it seems that red line is the ligand, right?
Yes.
Blue line is the protein.
Protein.
So the ligand seems to have moved quite a lot. So there's like
If-
two extra molecules. I do wonder, to superimpose the
Sorry.
brain structure after the recent election.
If you see here, it's starting from 0.8 and it's ending at 3.2. And if you see, the ligand is already a small molecule, and it can undergo various conformational changes because of its flexibility. And these flexibility of order of or changes, conformation changes in order of 1 to 3 angstrom are acceptable for such a small molecule in the binding pocket of a large molecule. So
Is it possible that one brain cell?
no. It's not possible. And there weren't. I haven't shared the slide where we actually do the superimposition of the dock and the structure. It has been gone through various conformational changes, but at the end, it actually restored the conformation quite similar to the docked one. So that is why we also concluded that these are non-significant conformational changes.
Wonderful presentation.
Thank you so much, sir.
I guess that this is short of time, maybe because you run only 29 seconds presentation, and at that time, everyone would like to go forward.
Exactly.
Extend a little bit to have a better
Exactly.
understanding of this compound. So it's only 20 seconds normal.
Actually, we are doing this also. We are going to run now 100 nanosecond.
How long it take for you for 20 seconds?
How
long it take for you?
The over 20 nanoseconds.
Yes.
It took us almost 15 days because of
Yeah, at that time.
exactly because of the facility
And
we have.
the corona time.
It's the peak time.
Yeah, exactly.
And we just actually did this study in a matter of. I submitted the paper within 15-20 days after that. So that is why I wanted to be short and crisp.
Yeah, I understand.
So that is why I chose these two brain children. The other were also proved to be really good inhibitors. They had really good blocking score and binding and everything.
Last thing, we performed the what is it? ADME before or after?
Yeah, this is also a question here. I did ADME for all of the 9 compounds, but I presented here only the 2 because of the shortage of the time. Exactly. So because I presented in the paper, in the publication, all of the information is available as supplementary.
We have time for 2 more questions if you have any. Okay, that is
Silvana, if I have. But now you have assays available, right? You can check it on actual viruses. Have you done that?
But sir, in my first we need to develop, I guess, in my opinion, the biochemical assay for just this molecule.
Biochemical assay you're already doing.
Actually, it's through NMR. First of all, we need to be sure with this one. Yeah, exactly. Or calorimetric or biophysical. In this case, because it's an enzyme, we can also do the calorimetric assay in addition to studies, the ligand binding studies. And that is how we can be sure that yes, the compound is acting through this. So we can proceed, because on viral assay, we will take the whole viral particle, we will grow the virus and then treat the whole viral particle with these compounds. And there are lots of compounds on there are lots of molecules on the surface of the molecule, the viral molecule, that can interact with these compounds and can lead to us to false positive or false negative. So that is why we are not going right now for the checking with the viral particle itself. We will instead go with the viral protein first.
Thank you, Dr. Louisa. From the organizing committee. Thank you so much.
So I'm organizing committee to organizing committee.
Okay, before we conclude the session, there's an announcement from the organizer that they have arranged a sightseeing tour for the foreign participants, delegates. The vehicles will depart at 6:30 sharp from the guest house kindly gather before time at the guest house reception. Thank you. And with that, we conclude the session. Thank you very much.